As Ceci said, I'm Thomas Chalaux. I'm a PhD at CEA and Kyoto Prefectural University under the direction of Olivier Evrard and Nakao Atsushi. And I worked with Sylvain Yon, who is a researcher at the IES in Paris. So, as we are late in time, just a short introduction of me. I did a bachelor in biology and ecology and a master degree in ecology and agronomy with a specialization in soil science, also known as pedology. And I started my PhD on the October 1st, 2021, on the impact of decontamination and recultivation of agricultural soils on sediment and radiocesium transfer in Fukushima rivers at the LSE in Paris, and I'm funded by the CEA. And I also was awarded by the GSPS to spend 1 year in Kyoto Prefectural University with Nakao Sensei. And we were awarded by the Iran grant to support our research on the Manodam Lake in Japan. So, as a presentation of the context, so as it was already said before, after the deposition, it was found that the majority of cesium was concentrated in the upper 5 centimeters of soil. And typhoons were shown to be the main driver of sediment transfer in the Fukushima region. As the Japanese government chose to actively reduce radioactivity, they published a decontamination guideline in December 2011 and a decontamination roadmap in January 2012. So it was quite fast after the accident. And the decontamination was focused on cropland, urban areas, and a 20-meter buffer zone of forest next to urban areas. Decontamination can be seen as 3 steps. So the 0 step is the undecontaminated cropland. And the first step is decontamination. So they remove the first 5 centimeters of soil. Then the remediation, when they add saprolite, so stone sand. And the last step is preparation for recultivation. So they mix all the first 30 centimeters of soil with the saprolite. Decontamination works were shown to be, so they leave the soil uncovered by vegetation, which may exert the soil erosion and so the sediment transfer to rivers and the cesium transfer to rivers. So the gap of time between 0 and 1 and 1 and 2 were shown to be highly sensitive. However, the vegetation grew very fast in the region, so it can protect soil from erosion. So here, the same location in April and then 3 months later this year. So we shall see that the vegetation is high. In Fukushima coastal catchment, the main land use are forests, about 80% of the surface, and croplands, about 10%. However, it was shown that despite their low relative abundance, croplands provide a major sediment source. So, and as they are the main focus of decontamination, they might impact the transfer. So it raises the following question: how does a fast and strong change of land use can modify radiocesium and sediment transfer at a river catchment scale? And to show you how do we work, I will present you the case study of the Hayama Lake, also known as the Manodam Lake, which was an early decontaminated catchment. So the Hayama Lake is located at the northeast west of Fukushima Daiichi nuclear power plant. And 3 cores were sampled in 2014 at the upstream, midstream, and downstream location of the lake. And the core sampling is a cooperative work between the IER, NIST, and LSE team. And a second campaign was done in 2021, and the midstream core and downstream core were sampled. And I focused on those 2 cores. And it's important to understand the temporal context of the catchment regarding the decontamination steps. And as we see, the decontamination started in June 2014 and ended in December 2016. And the first cores were sampled at the start of the decontamination work, and the second core in 2021. But when we look at the cumulated decontamination surface through time, we see that the majority of decontamination happened at the second part of 2016. So it can't be seen in the first course, but it will be seen in the second course. And as I said before, typhoons are the main driver for sediment transfer. So it's important to look at the temporality of typhoons in the period. And also when we look at the decontamination, we see that during the decontamination works, the majority of decontamination works occurred after the main typhoon of the period. So the first typhoon that will show the impact of decontamination is land. And the mixing of remediated soil started in 2019, but the add of saprolite is an in-between. So we will see how does the signature change through time. So to understand and date the sediment dynamic, we did a wide variety of measurements, like environmental DNA, geochemistry, grain size, organic matter and stable isotopes, radioactive isotopes, and also visible color spectrometry. These measurements have 2 objectives. First, dating the events, and then doing a fingerprinting approach. The eDNA is different because it's a more explorative measurements, and we will see how can we use this information to increase the discrimination of biological communities and understand more the change of land use. So as far as results, how do we dating the cores and the events? So this is a downstream core, and you can see that many layers were, you can see many layers. And when we add the cesium activity, you can see that the Fukushima accident is just below, about here. And we see different dynamics of radiocesium activity inside the core with the high plateau and a linear decrease and then lowest plateau. And maybe 3 periods can be associated to this region, like the abundant time, then the decontamination, and then the remediation. And to help to characterize these time period, we will use other analysis that was where was made, like grain size, organic matter, geochemistry, specific surface area, and XRD. And so it helps to classify the layers into these different regions. And with the rain events, we can better date the different events. Then it will be useful to do the sediment fingerprinting. So sediment fingerprinting approach helps to goal is to quantify the source contribution in target sediment. So when we speak about sources, it can be cropland, forest, subsoils, or decontamination steps. And the targets can be lake sediment core, but also lake deposit or suspended matters. So as a conclusion and perspective about the use of dating sediment cores, the IMA lake sampling at 2 different times is a unique opportunity to see how fast and strong change of land use can affect sediment dynamics in an agricultural catchment. And then it takes place into a more wide study that I call the Free Lake study, because 2 other cores were and will be sampled in 2 other lakes, in the Ogaki Lake and Yokokawa Dam Lake. And the interesting thing is that Ogaki Lake is in an lately decontaminated catchment. So it's barely the same condition as Ayama Lake, but the decontamination was later. And the Yokokawa Dam Lake is in a forest catchment. So it's like the Radiocesium transfer without human activity and decontamination works. So thank you for your attention.
Some question or comments or thank you.
The contamination process in the field seems to linearly decrease the radioactive sediment. Is that right?
This is what I also think. But right now I didn't have all the results, so I can't tell if it's really that. But what I can tell now, it seems like after 2016, we see a linear decrease of radioactivity, cesium activity. So it might be linked to decontamination. And also what I will do is to find, because the remediation with the iodosaprolite has a very specific signature. So when we see the apparition of decontamination, we will be able to date the decontamination period precisely. Is it clear?
You also analyze other element distribution by in the segment.
Yes, we did a lot of. I don't have everything here, but mainly we compare.
And you have the information that when the actual agriculture activity initiated in those areas?
Right now, no.
Thank you very much.
Thank you for your question.